nucleus is the control center of the cell. It's where we're going to find the DNA, the genetic information. So its job is to store and house that information. It protects it. We say it's the control center because that information regulates everything else inside of the cell. There's other stuff going on inside the nucleus. We make some things called RNA. That's the control mechanism. We house the DNA. Our genetic information is found in these strands called chromosomes. Most of the time when you're inside the nucleus, all that stuff is uncoiled and twisted. It's in this big fluid little bubble. And so we call it chromatin. Those two words become much more familiar to you in the next 2 units. We introduce the ideas now. Because our DNA isn't just one long strand. We have multiple strands inside of our nucleus. 23 pair, 46 copies. The nucleus itself is surrounded by an envelope with pores. That just means it's surrounded by a phospholipid barrier with holes in it. Because DNA is on the inside of the nucleus, but the place where we make the proteins, the place where we have to read the information, is outside the nucleus. So some information needs a way to get out of that nucleus, and it does so through these pores. Now, just like we called the fluid outside in the cell with all the little organelles cytoplasm, the fluid inside the nucleus with all of the DNA is called nucleoplasm. So cytoplasm is the fluid and organelles and proteins and things outside in the cell. Nucleoplasm is the grainy stuff all inside of the nucleus. We called it chromosomes and chromatin because when we first started having better microscopes and we were staining things with different colors, we noticed these darker stained things in the middle of the cell. We didn't know what they were, but they called them chromosomes. Chromo means color, zome just means body. They're these small little bodies inside the cell that had a color to them. So we call them chromosomes. And the name just stuck. A dark central portion of the nucleus, some textbooks refer to it as its own organelle. It's kind of deep in part of the nucleus. It's called the nucleolus. So say nucleus. Nucleus. That's the big organelle. Say nucleolus. Nucleolus. They're separate. So here's the nucleus. This darker central portion is called the nucleolus. And so here we're going to, all this grainy stuff is our DNA, all kinds of fluid in there. Here we're making things called RNA inside of the nucleolus. Look at this. This is an actual picture of DNA, or of a nucleus. People would have lost their minds if they could have seen this a century ago. And now we just take a picture and say, yeah, cool, here it is. Anyway, isn't that amazing? So we've used an electron microscope clearly here to get this outer picture of the nucleus, and we've learned recently there are little pores outside of it, little holes that allow our RNA to escape. Now it doesn't just escape by itself. They're in little containers called vaults, but we don't learn about vaults in here. But what's outside the nucleus? If inside the nucleus is where we build, nope, restart, Corey. If inside the nucleus is where we have our genetic information, how do we build a protein? Because DNA is the blueprint, it's the information, but somehow we have to get that information and build amino acids and stick them together. So where is that occurring? It's right outside the nucleus in what are called ribosomes. If you've ever learned the word ribosome before, high school, middle school, what have you, you probably learned them or saw them on the model as little circular dots outside the nucleus or out in the cytoplasm. There are actually two substances, two big globular proteins that pull apart and stick together when they go to do their function, but we just draw them as circles and models. So here's one. We kind of have a big glob here and a glob here. They're made up of both protein and RNA, but they read the information and form proteins. So we kind of just see it like this when we draw it in our textbooks. But if we were to zoom in, we could see that a globular protein, if you remember, is a tertiary protein. So it's a bunch of amino acids twisted and coiled together working to do a function. Directly outside of the nucleus and attached. So the nucleus is made up of a phospholipid bilayer because they're all endomembranous within a membrane. Attached to that phospholipid bilayer is a network of channels called the endoplasmic reticulum. Reticulum is an older word that means net or network, like a fishing net or something. Endoplasmic, endo means within, plasmic just means plasma. So within the network of the plasma, the endoplasmic reticulum is attached to the nuclear envelope, physically attached. It's part of it. The first portion of the endoplasmic reticulum is completely dotted with ribosomes. Now, so we don't have to write endoplasmic reticulum over and over and over and over. In biology, we just write ER. If you see ER, we know we're talking about endoplasmic reticulum. So the first big chunk of the ER, we call the rough ER because it looks rough. It's got ribosomes all over it, sprinkled all through it. So remind me, what is the job of a ribosome? What does a ribosome do? DNA stores genetic information. What does a ribosome do? It makes proteins. Ribosomes make proteins. So remember, I said that when you're studying for these big unit exams, there's a lot of information. And so you need to help yourself where you can. Kind of use process of elimination, use your context clues. Think, why do we call it rough ER? Because it's dotted with ribosomes. So even if we forgot what the ER does, you didn't have the slide in front of you, and I said, what does the rough ER do? You think, well, it's full of ribosomes. Ribosomes make proteins. So it must do something with proteins. Why else would it have all these ribosomes all over it? So we make the proteins in the ribosome. But we said there are 4 shapes we can bend a protein into, right? Either just the strand, leave it alone, make it go do its job. Secondary, tertiary, quaternary. Somebody has to be doing that, bending and folding. They don't just do it on their own. And so the rough ER allows us to fold these through some complicated mechanisms that we don't really care about. The rough ER is just folding them into shapes. I'm going to go to a different slide, but I'll come right back, I promise. So here's our rough ER, physically connected to our nuclear envelope. We make little proteins, they're dotted along here. The rough ER folds it. But there's a portion of the ER still attached through these channels, these little canals, that does not have ribosomes. It's on the outside of the endoplasmic reticulum, surrounding the nucleus completely. It does not have ribosomes, so we called it smooth ER. It didn't get too complicated there. One is rough, one is smooth. So it's not going to deal with proteins. It's going to deal with lipids and sugars. So our lipids and polysaccharides. Do you remember what a steroid is? What shape does a steroid have? 4 fused rings. It's a ring. So we have that structure. There's more to it, obviously, but that's just the basics here of what a lipid is. Steroids come in different forms, but their base structure is really just, there's other stuff. I'm just making that up, but their base structure is cholesterol, which is a lipid, right? So our steroids, some of our vitamins, estrogen, testosterone, are lipids essentially. So if you have a cell that's packed full of smooth ER, a bunch of it, you can probably assume it's making a bunch of steroids in some way or a bunch of lipids in some way. And so if we were to look at the cells inside of our internal sex organs, so if you look at testes or ovaries, we find a bunch of smooth ER. Well, why? Well, they're making testosterone, they're making your androgens, they're making estrogen, they're making progesterone. These are all just lipids, cholesterols. Rough ER folds proteins, smooth ER makes lipids and polysaccharides. They both transport materials to the very next organelle. So if we had our rough ER, smooth ER, it's still a system of channels, but it is not connected to the nuclear envelope. It's called the Golgi apparatus, or Golgi bodies, named after the person that found it, or looked at it and described it, last name of Golgi. It's a processing facility. So it's a network of channels. I can't draw, can I? And so here a little vesicle pinches off from our smooth ER full of a lipid or polysaccharide. It passes through each one of these faces, we call it. It passes through this side, this side, this side, this side, this side, this side, this side. As it passes through those faces, we add a sugar. The fancy word for adding a sugar is glycosylation. Glyco, which means sugar, and ylation, process of or adding to. So glycosylation is adding a sugar. Why would we waste time doing that? Remember, our cells all have that histocompatibility complex, that sugar fingerprint on the outside. What do we call it? Glycocalyx on the outside of our cells gives us that fingerprint, our cells to know what they are. These sugars act as sort of a GPS locator, or like an address label or something. And so they know where to go. We stick a sugar to a protein, stick it to a lipid, stick it to what have you, and it knows where it should go, where it should end up. Either stay within the cell or leave the cell and go on and do its other job. So we call it the processing facility. But there are two more things the Golgi does that gets forgotten a lot. One important one is plants make cellulose here. So memorize that. The Golgi is not just a processing facility that adds sugars, but plants make cellulose here. Do you remember what important structure is made up of cellulose inside of plants? Cell wall. I heard some people say the cell wall. And non-secretory cells make lysosomes. Now, we don't know what a lysosome is yet. That's our next slide. What is secretory? It's not a very nice word, is it? Secretory sounds kind of gross. Secrete. If you can squeeze things out of your cell, if you can release things, you're a secretory cell. When you release a hormone, you're secreting the hormone. If you're non-secretory, that means you're not releasing that stuff. Well, there's a lot that goes on inside the cell that we have to recycle or break down or get rid of. If you can't just spit it out, you got to deal with it inside. And so we make these lysosomes that can break things down. So non-secretory cells make lysosomes here because we're beginning to develop the idea, as we learn these definitions, that these endomembranous organelles are really just sections of phospholipids that have been pinched off and do something different. And so that's all it is these packets of phospholipids with a little bit of information in them that can do different stuff. And so when we get to I went the wrong way, when we get to the Golgi, to make a lysosome, you just pinch off a piece of the Golgi and on it goes. We don't name the faces in this class. Each of the faces have a different name. We call the side that's on the same portion the cis face, and the side that's on the opposite side the trans face. But we don't have to worry about naming them, but all your pictures put them in here. So lysosome. Zome means body. Lyso means to lyse or to cut. Loose body produced by the Golgi. But what does it do? What is the portion of the lysosome? A lysosome is full of enzymes or acids, things that have a very low pH. They break things down, is what they do. So your lysosomes can go and merge with something we don't need anymore, merge with the material that we don't want, and break it down. Now, most of the time we keep these enzymes in an inactive state. We don't want to break everything down until we need it. But lysosomes break down foreign aging material, break down old cell material. They allow us to recycle. Because remember, we learned back in chapter 2 and 3 that a lot of stuff inside the cell are just a bunch of carbon and hydrogen that we stick functional groups to do other stuff. So when we break things down, we're not just going to forget about that. Those are precious resources. We're going to recycle those bits and use them again for something else. Our white blood cells, our immune response, are also able to use these lysosomes to engulf pathogens, bad stuff, and kill them, or break them down, or try to get rid of them as best as they can. So remember, if you think lysosome, lice to cut, full of acid, then maybe you don't have to worry about memorizing all 4 of these bullet points if you just have in the back of your mind, lysosomes break things down. If you have some sort of question asking you about lysosome, you can use process of elimination. We okay with those so far? We had nucleus, rough and smooth ER, Golgi, lysosome. The reason we talk about peroxisome, it's not the most important in general biology, but we do a lab with peroxisomes. They're very similar to lysosomes. They have some enzymes. Most important, an enzyme called catalase, which we're going to learn when things end in a sugar, or things are a sugar, they end in OSE, right? Glucose, maltose, lactose. We're going to start learning, especially in the next unit, that if you're an enzyme, you end in ASE, ACE. And as we got better with naming, we didn't try to trick ourselves. So things like amylose is broken down by amylase. Lactose is broken down by lactase. Maltose, maltase. We got better at these types of things. Catalase is an old word for catalose, really, for hydrogen peroxide. These peroxisomes, if you think peroxide, produce H2O2, or hydrogen peroxide, as a byproduct. Hydrogen peroxide is very toxic to our cells. We cannot have it around for a while. It's dangerous. But look at what makes up H2O2. We can break it apart into 2 innocuous substances that we need, water and oxygen. And so catalase lets us break down that peroxisome. So why would we have this dangerous organelle inside of our bodies? Why would it make this byproduct if we have to worry about immediately breaking it down? Because it allows us to break down lipids. These peroxisomes let us break down lipids. Remember, we said lipids were nonpolar, and so they don't always want to react in the way we want them to react throughout the body. Or I shouldn't say want to, but they're more difficult to break down, and so we have these peroxisomes that allow us to degrade them, break them down. And so your peroxisomes, you're going to have a bunch hanging out in your liver, making bile salts. They let us break down our fats. If you're a plant, you can then convert those fatty acids into sugars. You can recycle things. So you can turn your oils into sugars. So peroxisome, make H2O2 as a byproduct. Let us break down fats. Let us break down lipids. So again, here's a picture. This is pretty cool. Here's a mitochondria off to the side here. Peroxisome fragment, probably breaking down some lipids, making oxygen and hydrogen or oxygen and water. We'll talk more about those in lab. The next big organelle, it's kind of a big group for a lot of things, are called vacuoles. I always learned vacuoles, vacuum, open space. They're made up of a phospholipid bilayer, but they're not found in all cells. They're a place to store stuff. If you're a little protist, sometimes you can use them to contract and digest things. But for the big place we're going to see a vacuole is inside of a plant cell. Plant cells have a giant vacuole right in the middle called a tonoplast. Plastid is just made up of a phospholipid bilayer, but a tonoplast. We call it the central vacuole, tonoplast, same thing. It's what holds and conserves our water. So I'm going to go to a picture. By our, I don't mean me, I don't mean you, I mean plants. I don't know why I said our. But holds and conserves and stores water in a plant. So they have this big old piece, this big old giant vacuole in the middle of their cell that holds water. Anybody ever forgotten to water a plant for a while? Kind of wilts. If you catch it, you can water it and it perks back up. Too far, it's just gone. It's dried out. But you can catch it sometimes. Water it, it perks back up. Like, good. Had that for a couple of years. Didn't want to lose it. There we go. The water that we're losing and adding, the reason it's wilting like that, is it's being stored inside the central vacuole. As we move into Unit 2, we'll start to learn about the rules of water and why it moves in and out. But right now, just know that it's stored in this vacuole. If the plant is out of water, it loses and loses and loses and loses and loses water until that plasma membrane begins to pull away from the cell wall and your plant wilts. Now, you lose too much water and it rips apart and there's no going back. Same thing's true if you overwater, these tonoplasts can burst. And then, of course, that also kills your plants. And so there's a fine line here. And of course, it all depends on what plant you're talking about. Every plant is different. But it's the central vacuole holding the water. And in plants, they can store some pigments, store waste, because if you're this big old giant thing, you're going to put it to more use than just holding water. You can allow it to store some of your stuff.